Good day, everyone. So we will be discussing about calcium and phosphate. And since this is your clinical chemistry laboratory subject, so we will just be focusing on the laboratory side of these electrolytes. So this will be our objectives. We'll not read it one by one na, so we'll just kindly check it na lang sa handout posted on your Quipper, and we'll proceed with the first one. So for calcium, take note that your calcium is the fifth most common element and it is the most, I think second most prevalent cation in the human body next to sodium. So, in terms of the specimen needed or preferred specimen for the determination of calcium, it depends on what we are trying to determine, whether it is your total calcium or your ionized calcium. Now, for your total calcium, the preferred specimen or sample would be either a serum or if plasma, then the anticoagulant that you need to use is a lithium heparin. Citrate, EDTA, or even your oxalate should not be used since it can interfere with the analysis. Remember the function or the action, rather, the action of your citrate and EDTA, the action of these anticoagulants, but they are more on the chelation of your calcium, chelation or binding of your calcium, and thus it is unacceptable for use since what we are trying to determine here is the level of your calcium, and thus it will affect your calcium determination. For ionized calcium, it must be collected anaerobically. Diba? When we say anaerobically, it should be without the presence of oxygen. Now, why? Uh, Any change in the pH could actually affect your calcium level. And with the presence of oxygen, as your oxygen number increases, it could also change the strength of your pH. As your pH increases, or meaning nagiging alkaline yung ating pH, it promotes increase in your protein binding, increase in your protein binding. Thus, in turn, because there is increased protein binding, then there would be an increase in your calcium level. Kasi we are looking at the ionized or your free calcium here. So, since nag-increase yung protein binding, meaning there will be a lot of calcium that will bind to your protein, kaya nagfo-falsely decrease yung ating ionized calcium level. And of course, vice versa, if there is acidosis or kung nagdi-decrease yung ating pH level, then mag-decrease din yung protein binding and then resulting to a false elevation in your free calcium levels. So that is why we need to make sure that your ionized calcium should be collected without the presence of oxygen. So paano po yan? It would be best to use your evacuated tube system. Di ba? It is a closed system, so meaning less likely to be contaminated with oxygen. The preferred sample for your ionized calcium would be your heparinized whole blood. But if wala po tayong heparinized whole blood, then serum sample is still acceptable. Just make sure not to use, ito po, not to use a liquid heparin since heparin concentration of 25 IU per ml decreases ionized calcium by about 3%. And 3% is already a very big number. Now, we could also measure calcium through the use of your urine sample, pero hindi siya yung random. Much preferred if it is a timed urine collection like your 24-hour urine collection. But just make sure that for this collection, it should be acidified with around 6 moles per liter of your hydrochloric acid. And it is approximately 1 ml of the acid. Approximately 1 ml of the acid is added for each 100 ml of your urine. Now, what is the purpose of adding hydrochloric acid in your urine? Dalawa po yung purpose niya. One, it could be as a preservative. And the other one is to prevent calcium salt precipitation. Let me just write it down. Prevent calcium salt precipitation. So that is why it is very important to add hydrochloric acid in your time urine na collection. So other sample considerations when determining your calcium are the following. Whole blood specimens should be analyzed within 15 to 30 minutes. If this is not possible, so what you need to do is to place it or kept it in ice. Because once your sample is placed on ice, it can be stable for at least 2 hours. Now, if the specimen cannot be analyzed, as in busy ka masyado, hindi mo agad mapoprocess yung inyong sample within an hour, then the preferred sample would be your serum. Now, hemolysis and delayed plasma or serum separation would lead to a decreased calcium concentration. Kaya always process your samples immediately. Because any samples, hindi lang naman po kay calcium, again, any samples upon prolonged standing ng inyong samples, may effect po talaga siya. There will be changes that is happening there in your sample, and that is why it is always preferred to immediately process them. Another, a decrease in your plasma protein concentration will result in a decrease in your total calcium because remember that around 40 to 50% of your calcium are bound to your proteins, and thus any changes in your protein concentration would also affect your calcium concentration. Next, recumbent posture. Posture could also decrease your calcium concentration. Samples should be drawn with the patient in a sitting position. Standing increases the total calcium concentration. Uh, uh, I think yung pwede natin tingnan dito is in terms of the changes in your plasma volume. Since every time we change our position, there's also changes in our plasma volume. And uh, as what I have researched, plasma volume change of about 3.4% happened if from lying down to sitting is around mga 14% if galing ka nakahiga tapos tatayo ka kaagad, and around mga 9% from sitting to standing. So that is why upon collection of your calcium sample, it would be best na to let your patient rest first before drawing the blood. Next, we have this one. Venous occlusions would increase your calcium, especially especially during blood collection also. As simple as fist clenching or forearm exercises done by your patient could also lead to false elevation of your ionized or free calcium levels. So this is basically on the hemodynamic changes lang po that will happen during these activities. So now we go to the laboratory methods of your calcium concentrations. So for total calcium concentration measurement, when we say total calcium kids, it involves measuring both your protein bound and your ionized calcium. Now, as what I have mentioned a while ago, diba, as what I have mentioned, since around 40 to 50% nakabind si calcium sa protein, especially your albumin, then any changes in your protein or albumin concentration could also affect the level of your calcium. So that is why, if your patient has hypoalbuminemia, it will lead to a misleadingly or false decrease of your total calcium level. So in order to kumbaga, in order to note this hypoalbuminemia, in order to really get an accurate result of the total calcium concentration of your patient, a correction must be made. And thus, as you can see here, this one, naka-box, this is your formula for the corrected total calcium level in cases of if the patient may hypoalbuminemia. So, first, you need to determine the albumin concentration of your patient, subtract it dun sa normal albumin normal reference range of your albumin. And then once you get that, add it to the total calcium measured and then multiply it by 0.8. And thus, whatever is the result, yan po yung ating i-report. So these are the different names of the methods used for total calcium measurement. So you have your orthocresylphthalate complex zone method, or otherwise known shortcut, your OCPC. Now, OCPC reacts with your calcium to form a purple color in an alkaline solution, and it is measured at this wavelength around 580 nm. Another important reagent content of your OCPC method is your 8-hydroxyquinoline, in which this specific substance prevent any interference of your magnesium. Next method is your arseniazo-3 dye method, wherein your arseniazo-3 will react with your calcium to form a calcium indicator complex, and this is measured at this wavelength at near 650 nm. Our reference method for the total calcium measurement is your AAS, or your atomic absorption spectrophotometry. And then we can also use this method, your ion selective electrode. So I guess are already familiar with these two, so no need to reiterate once again its principle. So let's proceed with the next. So this video would show you as to, at least you will be able to have an idea as to how it is being processed. So this is just an example kaya para at least ma-imagine nyo lang din and you get to appreciate how your electrolytes are being processed in the laboratory. So kindly watch this video.
Hello everyone. In this video tutorial, we'll see how to perform calcium test by CPC method using the kit manufactured by Anamol Laboratories Private Limited. First, let's have a look at the kit components. The kit contains one bottle of calcium CPC reagent, one bottle of calcium CPC diluent, and one bottle of calcium standard of 10 mg% concentration. The kit also contains the instructions for use. We recommend all the users to read the instructions for use thoroughly before using the test. Apart from this, you may require additional items such as clean and well calibrated pipette of 1000 microliter volume, clean and well calibrated pipette of 100 microliter volume, clean and dry tips of 1000 microliter volume, clean and dry tips of 100 microliter volume, tissue paper, clean and dry tubes in which the test is to be done, patient sample, and timer or stopwatch. Before we go ahead with the procedure, first let's pay attention to the preparation of working solution. The working solution can be prepared by mixing 1 volume of CPC reagent with 1 volume of diluent. Now, as per the procedure, we need to take 3 different tubes, one for blank, one for standard, and one for test. In the first tube of blank, we need to add 1000 microliter of working solution only. In the second tube of standard, we need to add 1000 microliter of working solution and 20 microliter of standard. In the third tube of test, we need to add 1000 microliter of working solution and 20 microliter of patient sample. And then we need to mix it and incubate for 5 minutes at room temperature. After the incubation, we need to aspirate the reaction mixture into the instrument and take the absorbance. To prepare 1000 microliter of working solution, we need to take 500 microliter of reagent and 500 microliter of diluent. So now we will prepare the working solution. To do this, first we will take 500 microliter of diluent without any air bubble. And then we will add it to the first tube which we have already labeled for the blank. Then we will take 500 microliter of the diluent without any air bubble and add to the second tube which we have labeled for the standard. Similarly, we will take 500 microliter of diluent and add to the third tube which we have already labeled for the test. Then we will change the tip and then we will take 500 microliter of the reagent, CPC reagent, and then we will add it to the first tube labeled for blank. Similarly, we will take the CPC reagent and add to the second tube labeled for standard, and so on for the sample also. Please note that while adding the CPC reagent, we are not mixing the components of the tube with the tip. Mix the reagents and now our working solution is ready. So now we will add the standard as well as the patient sample into the working solution. To do this, we will take 20 microliters of standard and wipe the outside of the tip with a tissue paper, and then add the standard to the second tube which we have already labeled for the standard and mix it thoroughly. Then we will change the tip and then we will take 20 microliters of patient sample. Again, wipe the outside of the tip with a tissue paper and add it to the third tube which we have already labeled for the test. Again, we will mix it thoroughly and then we will start the incubation for 5 minutes at room temperature. While the incubation is going on, make sure that you switch on the instrument and open the program for the CPC reagent and keep it ready to read the blank. Please refer to a separate video in which we have explained clearly how to program the calcium CPC reagent on any semi-automated biochemistry analyzer using the information provided in the instructions for use. So now the program is ready and now the program is showing the message to read the blank after the incubation is over. So once the incubation is complete, what we will do is we will directly take the tube of the blank and then aspirate the tube to read it. After a small lag time, the instrument has already calculated the absorbance of the blank. And now the instrument shows the message to read the standard. So now we will take our second tube, for which we have already prepared for the standard, and then aspirate this tube. Again, after a small lag time, the instrument is giving the message to rerun the standard. Now in this case, we have only prepared one tube for the standard. Hence, we will select the option No. As you can see, the instrument has calculated the absorbance of the standard and also calculated the factor. Now the instrument is giving the message to read the sample. So now we will take the third tube in which we have run the patient sample and then aspirate it. Again, after a small lag time, the instrument has already calculated the absorbance of the standard and also calculated the result. If you wish to run more patient samples, you can follow the exact same procedure. This is the correct way to perform calcium CPC test. Thank you, and we hope this video has been helpful for you.
Okay, so yun po. That is how you are going to process your sample, especially when you are doing manual, yung manual po na way. But the good thing about here in this video, kids, is that the machine that they are using is already a semi-automated one, in which actually we also have that in the school, sa laboratory po natin sa school. Not exactly the same machine, but same yung concept na we already have the probe, tapos pinapasip na lang po natin yung ating sample, and then instead of just giving us the absorbance, yung binibigay na na result dun sa ating machine is already the concentration. So, no need to compute manually for the concentration of your sample, since yun na nga, kino-compute na agad ng ating machine, and the result that is being given by the machine is already the final concentration of your sample. I hope you got to appreciate the whole process. Now, dun sa video, of course, we will not follow what is stated there in terms of how you are going to process. But again, technically, ganun po yung ginagawa natin. But of course, we are using a different one. So let us focus with the one that we are using in the lab, and that is your humalizer. So, this, ito po na manual na instruction, I also posted this in your Canva, so I hope you downloaded this one. So let's proceed. Now, for this method, kids, what we will be using is your CPC, your OCPC method still. And in terms of the method stated here, it's just the same as what I have stated a while ago, that your calcium ion will react with your OCPC in an alkaline medium and then forming a purple colored complex. And that, take note that the absorbance of your complex is directly proportional to the calcium concentration in your sample. So now let us check the contents. So we still have your buffer solution. Content of your buffer solution is your lysine buffer at pH 11.1. Then you have your sodium azide. So recall that the purpose of your sodium azide is just as a preservative, but careful with the use since sodium azide is harmful. While for your buffer, so recall lang din po natin yung purpose ni buffer, that buffers are solutions that can resist your pH, resist any pH changes upon the addition of either an acid or a basic component. So parang sila yung nag-neutralize. It is able to neutralize small amounts of added acid or base, thus maintaining your pH dun po sa ating solution. So next would be your color reagent. So as you can see, yung na-mention natin kaganina, meron po tayong 8-hydroxyquinoline. Again, going back to the purpose of this substance, it is to prevent any magnesium interference, since magnesium could affect the overall assay or process of our calcium determination. Then you have your OCPC, then you have your hydrochloric acid. Forgive me, kids, tried to search as to what is the purpose of hydrochloric acid there in the reagent, pero hindi ko po kasi nakita. So, let's leave it as it is na lang, ganun na lang. For the standard, so you have here specific, so you all know the purpose of your standard, and then andyan po yung specific concentrations for our standard. And then for the reagent preparation, what you need to do is to add your reagent to your buffer in equal volumes as required, mix and allow to stand for around 30 minutes at room temperature before use. Since, as you can see, ang haba po ng oras na ating hihintayin bago po natin magagamit yung ating working reagent. So that is why what I would usually instruct to the students is before everything else, before mag-start and before I would explain yung whole process, pinapamix ko na po kayo nito. Okay, para at least we could really maximize our time rather than just waiting 30 minutes and do nothing. Diba? Sige, next. For the reagent stability, so your reagent and standard are stable even after opening up to stated expiry date as long as it is stored around this temperature, and of course contamination should be avoided. Working reagent is stable for 7 days at refrigerator temp or for 3 days at room temperature. For your specimen, it's either your serum or heparinized plasma. Stability of serum is at around 10 days at 2 to 25 degrees Celsius. So now we proceed with your assay. So for your wavelength, you could either use 570 nm or 578 nm. And then for temperature, that is room temperature lang din, 20 to 25 degrees. And then for the measurement, it is measured against the reagent blank. And then this would be our pipetting scheme. So of course, for reagent blank, it is just your working reagent, nothing else to be added. For your sample or standard, syempre pag standard, yung standard natin na reagent bottle, yun yung ating idadagdag sa ating working reagent. While for sample, of course, that would be your sample. So, sample or standard is only 20 microliter. Working reagent is around 1000 microliter or 1 ml. So, mix and measure the absorbance of your sample and standard against the reagent blank within 5 to 30 minutes. So, since you are giving a range, so usually usually yung i-instruct ko sa mga students dyan is always dun sa pinaka-minimum. At least 5 minutes since 5 to 30 minutes. So, 5 minutes pa lang, pwede na talaga i-read. And then here, as you can see, kids ha, how the mo aning calculation kay murag pila na ka, pila na ka mga ingani ang ginadiscuss sa inyo ha. So, the formula is still the same. What differs is in terms of the multiplication factor that will be used. So, for your calcium, take note of the two units. You have your milligrams per DL and you have your millimoles per liter. For your mg per DL, your multiplication factor there is 8. For millimoles per liter, that would be 2. Again, always the same ang ating formula to get the concentration. That would be absorbance of your sample divided by absorbance of your standard. That is why dapat po talaga natin i-read the standard. So, you can see dun po sa video, always una yung blank, tapos yung standard, tapos kung may control, better mag-next control, saka pa ang inyong sample. Then, for the linearity of our assay, test is linear up to the concentration of 15 mg per DL or 3.75 millimoles per liter. If your sample has a higher concentration than that mentioned, then a dilution should be made 1:1 with the mixed with your distilled water. Then, syempre, once you get the result, since nag-dilute ka man, then you need to multiply the result by 2. And then, for the normal values of our sample, so ito lang din po. So, for your serum or plasma, it could be, kung mg per DL, that would be 8.1 to 10.4. For the millimoles per liter, that would be 2.02 to 2.60. What's next? Dito po tayo sa notes. Okay na yung quality control, okay na yung automation. So, dito po tayo sa notes. Contaminated glassware is the greatest source of error. So, disposable plasticware is recommended for the test. And then, test is not influenced by hemoglobin up to 200 mg per DL and bilirubin up to 20 mg per DL. As long as hindi magre-reach sa ganyan na amount, then no problem. But if ako yung tatanungin nyo kids, always, kung ako, better, wag na lang gamitin, embolize yung sample kasi you'll never know. So to really get an accurate result, dapat maganda din po talaga yung ating sample. Diba? Recall that most errors in the laboratory sa anong phase? Nasa pre-analytical phase. Next, dipemic and hemolytic samples require a sample blank. So by using the same pipetting scheme, mix 20 microliter of your sample with 1000 microliter of distilled water and measure the absorbance against distilled water. So sample blank has to be subtracted from the absorbance of your sample should be subtracted from absorbance of yung ginawa nyo po dito sa number 2. And then, buffer and standard contains sodium azide. So, this is what I mentioned also, na sodium azide is a preservative, but of course, do not swallow since sodium azide is harmful and it should avoid contact with skin and mucous membranes. So, yun po. This reference range, as you can see here, is the one placed in your bishop. And dito po kasi na reference range, naka-specify in terms of what we are looking at. If bata ba siya, or adult ba siya, if total ba siya, or ionized, if serum or plasma, or whole blood, or kahit nga yung 24-hour urine sample natin. So, be also familiar with this one. Okay, so now, we are done with our calcium. So, let's proceed with our phosphate. So, if you could still recall, iba parang tandem si calcium and si phosphate. Yung relationship nila, parang sodium and potassium din. Na if na somehow may interchange or exchange sa kanila. If mag-increase yung isa or mag-decrease yung isa, then there is interchange with their levels and their as to kung saan sila, yung position nila. Kasi 'di ba extra and intra din yan. So it's the same way with calcium and phosphate. Parang pair din ito sila. Now, for the specimen, serum is preferred for the phosphate determination, or if wala it's not possible for us to get a serum sample, then you can make use of plasma with the anticoagulant to be used na si lithium heparin pa rin. Oxalate, citrate, or EDTA can interfere with the analysis. And of course, as usual, hemolysis should be avoided. Circulating phosphate levels are subject to circadian rhythm. Diba, when we say circadian rhythm, uh, may variation yung kanyang concentration depende sa time of the day. So, for phosphate, highest is in the late morning and its lowest concentration is in the evening. And then we could also collect a 24-hour urine sample if we want to measure phosphate sa urine. So, yung best or recommended is a 24-hour urine sample. So, other specimen or sample considerations for phosphate. Phosphate concentrations in plasma or serum are increased by prolonged storage with cells at room temperature of 37 degrees Celsius. So, again, as what I have emphasized a while ago, it would always be best to process your samples immediately rather than letting it stay lang, nakastay put lang dyan for a longer period of time. Inorganic phosphate increases by 4 to 5 mg per dl or 1.29 to 1.61 millimoles per liter per day if your sample is hemolyzed, even if it is stored at 4 degrees Celsius, and more rapidly at room temperature or at 37 degrees Celsius. Phosphate is stable in separated serum for days at 4 degrees Celsius and for months when frozen, provided evaporation is prevented. So for the laboratory method, uh, dalawa po yung known or ginagamit na method for your phosphate. One is this. It involves the formation of your ammonium phosphomolybdate complex. And actually, meron po itong specific na name, ito po na method with the use of your ammonium and with the formation or end product na si ammonium phosphomolybdate. And that is your, kindly take note, your Fisk-Subarow. This is what we call your Fisk-Subarow method. Now, this ammonium phosphomolybdate product can actually be measured by either UV at 340 nm or by reduction method, reduce natin siya to become your molybdenum blue, which is read between 600 and 700 nanometer. Now, how do we reduce your ammonium phosphomolybdate into molybdenum blue? Marami po tayong mga reducing agents na pwedeng gagamitin. So, let me just write down what are these reducing agents that can be used. I'm not sure if nasa PowerPoint ko siya. So, let me just mention. You have your pictol. You have your elon. You have your cenidine. Hope I pronounced it correctly. And you also have your ascorbic acid. Now, for your pictol, this is also known as your amino-naphthol sulfonic acid. Let me just write it down here above. Amino-naphthol sulfonic acid. Again, this is your pictol. While for your elon, this is also known as your methyl amino phenol. Yun. I hope mabasa gihapon siya. Kaya I'm not really that, wala pa ko naanan o gamit sa akong pen for writing it here. So, ayun, again, These are the reducing agents that can be used in order to reduce your ammonium phosphomolybdate to become your molybdenum blue. And this molybdenum blue would be the one that we will be kumbaga measuring in order to get the concentration of your phosphate. So formation of your phosphomolybdate complex is pH dependent. Thus, we need to maintain your pH at a certain level. Rate of its formation is influenced also by protein concentration. So this is the second method for your phosphate determination. It is an enzymatic method in which we will be using or through the we will be using these enzymes present sa kanyang principle or reaction principle. It is catalyzed by glycogen phosphorylase, phosphoglucomutase, and G6PD. And what we are trying to measure here is the presence or formation of your NADPH. NADPH produced can then be quantitated either fluorometrically or spectrophotometrically. So now we proceed with our package insert with regards to your phosphate, the measurement. So here in your phosphorus determination, we try to look at it even if wala pong nakasulat na specific name sa method. If we try to read the method here, since it mentioned that phosphate will react with your molybdate in a strong acidic medium to form a complex. Basically, we are referring to your Fisk-Subaru method, and that the absorbance of this complex in the near UV is directly proportional to the phosphate concentration. You have your reaction principle here. Bale, if we try to look at itong mga chemical formula, so the first one, your H3PO4, is actually referring to your phosphoric acid reacted with your molybdenum. Yan po ito. This is your molybdenum plus your hydrogen ions, and then our product would be this one is your ammonium phosphomolybdate, and then releasing around 36 na water. Yan po. And then, looking at the reagents, here in your reagents, so ayun si ammonium phosphomolybdate. Then you have your sulfuric acid. Dalawa ang purpose ni sulfuric acid dito. It could also act as a preservative, and it can also be your precipitating agent. Then for detergent, try to search the purpose of detergent being added in different assay methods. And ang isa sa nakita ko po is it could actually act as a buffer. And then you have your activators and stabilizers. So here is your standard with the specific concentration of your phosphorus. And then for reagent preparation, wala po tayong mix na gagawin. Reagent and standard are ready for use. Stability, your reagents are stable even after opening up to the stated expiry date as long as you will store it around 2 to 25 degrees Celsius and no contamination. For specimen, so you can see here, sorry for the long pause, everyone. So, going back, as you can see here, since ang nakasulat po dito na specimen is your serum, so di po tayo pwede gagamit ng nung mga may anticoagulants or plasma. Again, the assay here is designated to just use your serum sample. So let us follow that one. Now, as you can see, nakasulat din pala, plasma must not be used. Anticoagulants may cause false results. Then you already have here stability of your serum samples. Again, take note, depende kasi talaga yan siya sa test kids. na whether or not pwede tayo gagamit ng plasma or hindi. Just like your machines. Actually, nag-vary din po yung mga requirement ng machines natin. Kasi there are some machines like for example, your uh, let us say your blood gas analysis. May iba pong mga machine kasi 'di ba what yung tinuturo namin sa inyo is to use your green top tube. But then there are some machines that would accept also your lavender top. And well and good kung ganun kasi mas common naman din si lavender top and then what if 'di ba the patient may CBC na request, then you do not need to collect a lot of blood samples and place it in different tubes since pwede naman gagamitin si EDTA tube. So mga ganun lang din. That is why it is really very important to read your package inserts. Next, now we go to your assay for wavelength. We could either use 340 or 334, temperature, room temp lang, and then against reagent blank. And then this is our pipetting scheme. So, reagent blank, 1000 microliter, sample or standard, 10 microliter lang, mixed with 1000 microliter of your reagent. And then, yung ating incubation time is just at least 1 minute at room temperature. Then, measure the absorbance against your reagent blank. And that, yung ginawa nating mix, yung mix sample and reagent na, stable yan siya for up to 60 minutes. So, kunwari, na-mix nyo na, tapos may ginawa kayong sideline, and then di nyo napansin na sobra na 1 minute. So again, you can still make use of that mixed na sample and reagent as long as hindi siya lalampas ng 60 minutes. And then here for the calculation, again, 2 units, milligrams per DL, millimoles per liter, and then our factor, 10 and 3.2. So the linearity of our test here is that it can measure phosphorus concentration up to 20 or 6.4. And as usual, if sobra yung concentration ni phosphorus dyan, then you need to dilute 1:1 with distilled water. Then result should be multiplied by 2. So that would be all for the package insert. Kindly double check lang kids ha, dun po sa ating package insert na ipo-post ko din sa Canvas na pwede nyo ma-download. Kasi baka nakat ko lang din, hindi ko rin napansin. Di ba nakita nyo walang nakastate na uh, reference range. So, kindly double check na lang din po dun kids. Because the reference range here that I have posted is sa Bishop na book. And depende siya. Serum sample lang din yung ating tinitingnan dito. And then, naka-age bracket pa din. For neonates, iba. Then you have child of less than 15 years old. You have the adult. And then you also have the result, the reference range rather, for your 24-hour urine sample. Okay, so that would be all for our discussion for today. Thank you for listening, everyone. And if you have any questions, please do not hesitate to place it sa baba ng video kung saan kayo pwede makakomment. So, have a nice day po and thank you.